Richard Marino. Okay, so today I'm going to be talking to you about the regulation of sperm maturation in the germinal epithelium. So first slide I have here is just an overview of sperm production. Sperm are produced in the testes. The initial stages occur within the testes and progress to the epididymis, where the developing gametes mature and are stored until ejaculation. The seminiferous tubules of the testes are the starting point of the process, where spermatogonial stem cells adjacent to the inner tubule wall divide into a centripetal direction, beginning at the walls and proceeding into the innermost part, which is the lumen, to produce immature sperm. The maturation process actually occurs in the epididymis, and the location of sperm production is important because spermatogenesis requires lower temperatures to produce viable sperm, hence why the testes and the scrotum are located outside of the body. The whole process takes about 74 days, where the testes produce 200 to 300 million spermatozoa daily, but only about half of them become viable sperm. It can be broken up into stages: spermatocytogenesis, which is mitosis. It's the male form of gametocytogenesis and results in the formation of spermatocytes processing half the normal complement of genetic material. And then we have spermatidogenesis, which is meiosis 1 and 2. This is where the creation of spermatids from secondary spermatocytes occurs. And then we have spermiogenesis. This is where spermatids begin to form a tail by growing microtubules on one of the centrioles, which then turns into the basal body, becoming motile. The next slide I have here are of the role of Sertoli cells in this process. They extend from the basement membrane to the lumen of the seminiferous tubule, and they serve many different functions during spermatogenesis. These include maintaining the environment necessary for development and maturation. They secrete substances initiating meiosis. They secrete supporting testicular fluid, secreting ABP, which is androgen binding protein, which concentrates the testosterone in close proximity to the developing gametes. They secrete hormones affecting the pituitary gland, and hence the control of spermatogenesis. They phagocytose residual cytoplasm left over from spermiogenesis. They secrete anti-Müllerian hormone, which causes deterioration of the Müllerian duct. They protect spermatids from the immune system of the male, and they contribute to the spermatogonial stem cell niche. And here I have a little image depicting where the Sertoli cell actually is in this process. The next slide I have here are some factors that influence sperm maturation. They're sensitive to fluctuations in the environment, particularly hormones and temperature. Testosterone is required to maintain the process of sperm maturation. This is achieved via the binding of testosterone by ABP, again that's androgen binding protein, present in the seminiferous tubule. And the testosterone is produced by Leydig cells. There are temperature sensitivities, of course. The seminiferous epithelium is sensitive to elevated temperatures in humans, and the testes are, like I mentioned before, the testes are located outside of the body in the scrotum at a maintained temperature of 2 degrees Celsius below the normal body temperature. This is achieved by the regulation of blood flow and positioning towards and away from the heat of the body by the semisteric muscle and the dartos smooth muscle in the scrotum. Next, we have the thermal exchange between the testicular, arterial, and venous blood streams. There are specialized anatomic arrangements that consist of 2 zones of coiling along the internal spermatic artery. This arrangement prolongs the time of contact and the thermal exchange between the testicular, arterial, and venous blood streams. And some other factors that influence sperm maturation are dietary deficiencies, anabolic steroids, metals, X-ray exposure, dioxin, alcohol, and infectious diseases can all affect the rate of spermatogenesis. Next slide, I have some factors that contribute to the hormonal regulation of sperm maturation. Here, hormonal control of spermatogenesis varies among species. In humans specifically, the mechanism is really not completely understood. It is known that the initiation of this process occurs at puberty due to the interaction of the hypothalamus, the pituitary gland, and the Leydig cells. If the pituitary gland is removed, spermatogenesis can still be initiated by follicle-stimulating hormone, which is FSH, and testosterone. And in contrast to the FSH hormone, LH, which is luteinizing hormone, appears to have little role in spermatogenesis outside of inducing gonadal testosterone production. And a little bit more on the FSH, or the follicle-stimulating hormone. This specific hormone stimulates the production of androgen-binding protein by Sertoli cells and the formation of the blood testis barrier. ABP, androgen-binding protein, is essential to concentrating testosterone levels high enough to initiate and maintain the process of sperm maturation. The next bullet point I have here, testosterone levels vary based on location in the body and the time of day. Intratesticular testosterone levels are 20 to 100 or 50 to 200 times greater than the concentration found in the blood. The testosterone follows a circadian rhythm with higher production in the morning, which explains why many males have morning erections. FSH may initiate the sequestering of testosterone in the testes, but once developed, testosterone is only required to maintain the process of spermatogenesis. Increasing levels of FSH will increase the production of spermatozoa by preventing the apoptosis of type A spermatogonia. And then inhibin, which is another hormone, decreases the levels of FSH. Next, I have another bullet point on the Sertoli cells. They mediate parts of spermatogenesis through hormone production. They produce the hormones estradiol and inhibin. And Leydig cells are also capable of producing estradiol in addition to their main product of testosterone. And estrogen, believe it or not, does play a role also. It's essential for spermatogenesis. However, a man with estrogen insensitivity syndrome was found to produce sperm with abnormal sperm count, but abnormally low sperm viability. Well, sorry, a normal sperm count with abnormally low sperm viability. Levels of estrogen that are too high can be detrimental to spermatogenesis due to suppression of gonadotropin secretion and by extension of intratesticular testosterone production. And the last and final slide, I have some disorders of this sperm maturation process. We have here something called oligospermia. This is something that is characterized by semen with a low concentration of sperm and is also a common finding in male infertility. The next point I have here is asthenozoospermia, which is reduced sperm motility. Next, teratozoospermia, which is reduced percentage of sperm with normal morphology. And combinations of these disorders are quite common. And as you can see on that bullet point, we have a very, very long name, oligoasthenoteratozoospermia, which is everything combined, or OAT syndrome for short. And that last bullet point there, we have a zoospermia, which is a very severe clinical phenotype because with this condition, there are no sperm found in the ejaculate.